Thank you, Giacomo, for taking the time to chat with me. I really appreciate it. I know that you're busy, and yeah, I look forward to just chatting more about our collaboration and some of the amazing things that your company and you are doing. So, could we start with maybe could you describe what Kinetic River does in the general sense?
Sure. Yeah, so Kinetic River is a company dedicated to the development of cutting-edge flow cytometry instrumentation and other optical instruments for mainly the life sciences, but we also do projects outside of the life sciences in optics-based diagnostics. So optics certainly is a unifying thread, but the main thrust of our work is all in flow cytometry and cell analysis.
Okay. And maybe for people that are not familiar with the technology, could you describe flow cytometry?
Sure. So flow cytometry is a discipline, a branch of science that studies cells, and it does so by funneling the cells in a liquid suspension in some kind of a liquid matrix, typically a water or water-based solution, at very high speed through a nozzle, so that the cells come out in single file, where they're interrogated by one or more laser beams. So flow cytometry is a way to analyze a vast number of cells, hundreds of thousands or millions of cells in a typical experiment, and extract multiple parametric information from them, mainly having to do with their function. So analyzing, identifying different cells on the basis of their protein makeup, their expression of antigens, and their nuclear conformation.
Great. Thank you for that. What would be the difference between your technology and what's currently out there from other vendors? And then you can go as specific as you want, but just in general.
Yeah, so flow cytometry has been around for many decades. It's a well-established branch of life science tools, life science instrumentation and practices. There's always new things that are being developed in order to enhance the capability of flow cytometry to study and analyze the samples. Where our technologies come in is in the extraction of deeper and richer information from each cell. And so as an example, flow cytometry typically uses fluorescence in order to label cells and identify cells on the basis of their antigen expression. Fluorescence is typically measured simply as an intensity and separated spectrally. So if you have blue fluorescence that identifies a certain kind of label or dye, and green fluorescence comes out of a different label or dye. And so spectrally, you can separate those two and use them to label different functions of a cell, and therefore identify different kinds of cells. What the technology that we've been developing over the last 8-9 years comes in is that we not only analyze the spectral characteristics of the fluorophores, of the fluorescence that comes out of each label, but we also analyze the lifetime characteristics. So very, very highly time resolved in a very, very short time scale, time resolved characteristics of the decay of the fluorescence from each fluorophore. And by using that additional information, which isn't available from traditional or conventional flow cytometers, we are able to extract more information, both from the labels that one can attach to the cell to identify the cell, and from components of the cell, molecules that are already inherent in the cell. That's called autofluorescence. And so by looking at the lifetime of the fluorescence from these different components, you can actually get more information and be able to discriminate cells more easily and more reliably and in a much more powerful way than is currently available and possible with other tools.
Okay, interesting. And I was going to ask you about label-free studies, but so basically label-free studies would be in addition to what you would do with actually using fluorescent markers.
Well, that's correct. Yeah. So fluorescent markers are typically referred to as labels. So they're
right.
You can analogize them to like a flag that you plant on a cell to identify the cell as one kind or another. If you have the ability to, and you can extract information from a cell without having to label it, that would be a label-free approach. And label-free approaches are very attractive in some cases, because just the fact of attaching labels onto cells changes the cells themselves. And so if you want to have a pristine analysis of the cells without having to change them or alter them in any way, if you can perform a label-free assay, label-free analysis, that you have a leg up. And so the fluorescence lifetime of autofluorescence is simply the analysis of the fluorescence that just naturally comes out of what's already inside the cell. So you don't have to label the cells. It is label-free analysis. And that's one of the things that our technologies provide.
And I think in one of the conversations that you and I had before, you were mentioning just the process of labeling can be just quite a time-intensive process. So that's also attractive in that sense.
That's correct. Yeah. So labeling involves several steps. It involves labels, which can be expensive. In fact, are typically in a flow cytometry laboratory, buying the monoclonal antibodies that are conjugated to the labels that are used for identifying cells is by far the biggest operating expense of any laboratory in the life science. It's just buying the reagents that enable you to perform the assays. So if you can extract information without having to label the cells, that could result in huge savings for certain kinds of assays. Okay. So there's that too. And again, there's, as you said, there's the burden of actually having to perform the incubation and making sure that there's no cross-contamination between the various labels that you use, and all of that is certainly a concern.
Okay, and maybe just to clarify, so you still couldn't do the same type of studies without labels that you would do with labels, but you could combine them and try to analyze specific things without labels. Would that be accurate?
Yeah. So let's say that we are actually we received a grant from the NIH to explore the possibility of using a label-free approach to get information about cells that you could get with labels, but if you are able to get it without labels, it will be much better.
Right.
And so we have actually completed that project, and the results that we obtained were very promising. There's more work to be done. It's not a slam dunk. It's actually a fairly complicated and difficult approach that we have chosen to do. But the results are very encouraging in that you could, for instance, be able to, you could distinguish cancer cells from normal cells on the basis of their molecular machinery. And what we've shown with our project is that you can separate the profile of a cancer cell from that of a non-cancer cell just on the basis of a label-free assay. Now, that's been done in microscopy already for quite some years. And the difficulty of doing it in flow, which is the whole basis of our technology, is that in flow, you're analyzing vast numbers of cells very fast. And so you don't have the luxury of having fixed cells on a microscope slide and be able to make them sit under the microscope as long as you want. In flow, the whole deal is you run your sample very quickly and you analyze a vast number of cells so you can have good statistics on a large population.
Okay, well, that's awesome. Maybe now that you talk about that, could you describe, you mentioned an application about finding cancerous cells. Could you describe another application where your technology and flow cytometry in general is useful and is used? And I don't know if there's anything about COVID-19 that would be useful to mention, because I mean, I know that that's in everybody's minds.
Yeah, so we're actually currently submitting a proposal to the NSF, the National Science Foundation, for a rapid diagnostic on COVID-19 that is based on flow cytometry. I can't say very much about it because it's very much at the early stages, and we are just finalizing the proposal at this point. But it is an interesting, a very interesting opportunity, because if you could have a rapid way of telling whether somebody is currently infected, not whether they had the disease at some point in time, which is also important, but whether they have the disease right now, that is perhaps more sensitive, or at least much more easily deployable. If it's very, very cheap, very cost effective, that could change the dynamics of testing altogether. Because right now the problem is that, yeah, you can do a PCR and it takes a while and it's quite expensive. So the cost that you're hearing from people who just want to get tested, if they're not covered by insurance, is like $100 a test. And
Right.
so if you can provide a test that's like single digit dollars, that's a game changer. That will be a game changer.
Wow. That would be, yeah, that would be incredible. Um, One of the things that I wanted to ask you about was about the Potomac. So that's kind of your flagship instrument. And one of the things that we mentioned in the case study that we together published was the ability to swap lasers. So I just wanted to specifically talk about that because that seems like it just gives you and your customers just tremendous flexibility. And together with that question, I know that we talk about deep UV lasers. So just wanted to get some of your comments about that just the ability of the Potomac system to be able to swap lasers.
Sure. Yeah. So as you said, the Potomac is our flagship product, and the best way to think about it is you can find instruments on the market where you can pick different lasers that you can put on the system, but you're choosing from a fixed menu. So you can say, well, I want a 488. Okay, so a 48 nanometer, that's in the menu, and then they offer a 640, and they offer a 405. And they may offer a 355 if you're lucky. So there's a relatively small set of lasers that typically manufacturers offer as a standard set of options. What we do differently is that we don't pre-assume for the customer what their application is going to need. And we ask them what kind of wavelengths would they prefer and would they ideally like to have on their system. And we simply build and redesign our Potomac in order to fulfill those needs. Mm-hmm. So every Potomac that we build is based on the same overall architecture, but we tailor it specifically for the needs of a customer. So for example, the one that we finished this year and we delivered to the National Research Council in Naples, Italy, this was a very unique system. It was based on essentially all UV excitation. So starting at 405, which is just on the edge of the UV, it's violet, it's just barely indivisible, and 375, which is a UV, and then 266, which is deep UV. And the customer had expressed a desire to have this range of very unusual excitation sources for flow cytometry because of the type of samples that they run. They're interested in doing water quality measurements. And so they are using these kinds of lasers to excite samples that might contain bacteria, contaminants, or algae, algal cells. And so these lasers allow them to do a label-free analysis of their water samples very rapidly. And so you just can't find an instrument like the one that they wanted on the market. And so we built it for them.
Wow, and that is a kind of a good segue for my next question, which was going to be who's your customer? So you mentioned research labs, and in this case, Italy. But can you describe like what's your target customer for your technology?
Yeah, no, that's a very good question. So for the Potomac specifically, because it's a fully customizable, fully tailor-made flow cytometer, the typical customer is a user who's ill-served by a cookie-cutter approach. So one of our earliest customers was Bill Telford at the National Institutes of Health at the National Cancer Institute. And he ordered a Potomac that was also customized specifically for his needs. And we made it possible for him to couple in to the Potomac any outside laser that he felt he wanted to use. And so he's been known in the community. He's been one of the pioneers of using exotic excitation sources in flow cytometry. And he needed an instrument that allowed that. And so we made it possible for him to have this instrument where he can couple in pulse laser, can couple in white light supercontinuum lasers and other types of lasers, so that he could explore different applications. And with the National Research Council in Naples, Italy, again, it's a similar idea where the 266 laser is actually external. And so you can couple that light in, and we made it possible for the excitation arm to accommodate deep UV wavelengths, which are rather difficult to handle, and most cytometers can't, in fact, handle that kind of.
And just taking a step back, so the reason that somebody would want to use different wavelengths is to be able to extract different information from cells. And
yeah, right.
Okay.
So different wavelengths can help you excite different labels if that's one of the things you're after. But also, as I mentioned, you can excite autofluorescence directly, and then you don't have to go through the step of labeling the cell at all.
Right. Okay. The next question was about our collaboration. So last year, I want to say last year or early this year, you and I had our first conversation. You had existing software and you needed some help basically taking it from where it was to actually being able to ship a new version of the software that actually goes hand in hand with the Potomac system. So I want to get your thoughts on the collaboration, specifically about the Panama software, and I may have some follow-up questions depending on your answer.
Sure. Yeah. So as you said, we started engaging JKI earlier this year to look at ways to develop an infrastructure, a software infrastructure that we could use in a variety of ways. And one of the ways that we wanted to have this software be able to address, one of the things that we wanted the software to be able to address is the ability to collect, if you will, conventional flow cytometry information, flow cytometry measurements from an instrument like the Potomac. At the same time, we also wanted to be able to collect information from a fluorescence lifetime type instrument like our Danubes and our Arnos and Colorados. And so a very different kind of underlying structure, but the software, the user interface needed to be essentially, very similar. So we have different types of hardware underlying in order to support the different timescales of digitization, of sampling. We have different requirements in terms of the storage of data, but we wanted a unifying user interface that we knew what we wanted because we had seen what it's on commercial instruments. We've developed our own custom UIs just for lab use, and we wanted to have something that could be deployed to a customer and also make our life easier in the lab. And so what JKI did for us is came up with a very robust software interface that we could use for both purposes. And then it was just tailored for the different applications, again, with the differences being that some of our instruments require much higher, much more finely sliced time-resolved information than a typical conventional cytometer.
And what would you say is different about the Panama software versus what you can get out there from another vendor?
Well, so each vendor typically provides their own software, all right, for running on their machines. You can get a copy of the software, but it's not going to do you much good if it's decoupled from the instrument itself. So we really, you really couldn't get a third-party software that's ready-made that you can just slap onto our machines. Right. So we knew that we had to make our own. We had already our own software for internal use, but like I said, we wanted something that was robust and customer-facing, and that's really where your team came in and helped us make that possible.
Right, and from the perspective of your customer, what were you trying to give them that existing software that they might be using for other instruments couldn't give them?
So, well, a couple of things, right? So we wanted to make sure that all the boxes were checked. In other words, people are used to, like, just like if you're a Mac or PC user, you're used to Microsoft Office, right? So you know your Microsoft Word and Excel and PowerPoint, and you're used to those kinds of looks and feels.
Right.
And so you don't want to have something that is like completely out of left field, because then they have to retrain themselves.
Right.
So we want it to be consistent with best practices in the industry, so that if somebody is a flow cytometrist and he or she is using an instrument, they don't have to like, okay, what the heck is this? You know, they want to have some familiarity with the way the data is displayed, the way the controls are arranged, and so forth. But we also didn't want to limit ourselves to what had been done in the past, because we had some features that we thought would be advantageous and better for the user. So we incorporated into the Panama software not only like the standard aspects of what you might find on a Diva software from BD, or other CAN softwares from different manufacturers, but we also wanted to give a deeper set of capabilities. And so without burdening the user with unnecessary under the hood information, we still wanted to give them a richer set of information that they could use.
Right, so striking a balance between showing enough information and giving enough knobs and things for the user to configure without just having everything in the main UI, so that it's too much, right?
Yeah, so one of the things that, from prior software examples that we were familiar with was, well, just the lack of ease of use, right? So the fact that, if you want to see a particular combination of parameters displayed in a scattergram, in a two-dimensional plot, you had to select them and pull them up, right? You had to actually create that window.
I see.
There's no reason to create that window if you have all the windows already possibly they can be displayed already available on a second page. You just turn to that page and there they all are. And so we made it easy for the user to just go and look, and they don't have to look all the time, but if they want to look, it's already there. They don't have to make it up. So that was one of the principles that we used, is we wanted to have the user focus on what they want to do and not what they shouldn't have to do.
Right. And maybe one more question about the collaboration between the two companies. So is there anything that you would say you were able to achieve with JKI that was either faster or better that you can comment on?
So I think what I was most satisfied with in terms of the collaboration was knowing that the robustness of the tool was as high as can be expected given the software infrastructure, the underlying LabVIEW framework that is provided, and the fact that it runs on a Windows machine, which also, they have their limitations. It's not a real-time instrument. But given those constraints, we are confident that our software is as good as anything else that you can see out there. In other words, that it handles memory as well as anything else. It handles all the input and output, the data transfer, and it performs very robustly, which is really what the user wants to have. They want to be stuck, dealing with error messages and things crashing, right? And so we knew we could get performance out of the hardware, and so also, JKI helped with identifying some hardware that was better suited for our purposes. But we wanted to make sure that all the housekeeping, the really under the hood stuff, was taken care of properly. That's one of the things that we knew we got.
Right. No, that's great. And from our perspective, the collaboration has been success, and your technology and your expertise coupled with what we know how to do, which is software, being able to do hardware integration. I think it was the perfect combination. I'm really curious about asking about the history of Kinetic River as a company, and I know that's tied with your personal experience. So I know that you So you have a bachelor's and master's from Harvard, then you went into Stanford to do a PhD in applied physics. So I'm just curious about the history of how Kinetic River got started.
Well, so before I started Kinetic River, I had been working at Abbott Laboratories for more than 6 years. And that's really where I got my deep dive into cell analysis. So I was in charge of a research group that developed hematology analyzers. And so we were developing advanced technology having to do with analyzing blood cells for routine diagnostics. So flow cytometry and hematology are very, very much cousins, right? First cousins. The hematology analyzers that we worked on when I was at Abbott are flow cytometers. They're just flow cytometers by a different name, and they have different purposes and different profiles, if you will, or specialties. But that's what they do. They do exactly the same thing. They funnel cells into single file very rapidly, and they analyze them with laser beams. And so that's really where I got excited and passionate about this technology and this approach of running cells at single file at highway speeds through a tiny nozzle that's microns in size, and being able to do that cell after cell after cell for millions of cells and extract a vast amount of information from each of them very reliably. And so, when I started Kinetic River, I wanted to be on my own. I wanted to have, my own path rather than being tied to a large company and, follow their vision. I wanted to have my own vision and follow that. And so I started the Kinetic River with the idea of bringing to the market some technologies in flow cytometry specifically that I felt would be difficult to bring to bear, to bring to reality under the umbrella of a large company.
I see. Was it and just curious, was it a difficult decision to jump off of a big ship into your own small boat at the moment?
It wasn't as difficult as staying on the boat once I did jump.
Okay, that's fair.
But
that was 2010, if I'm not mistaken.
Yeah. So the company was incorporated in 2010. I left my Abbott job in mid of 2011. So we've been in business for about 10 years now.
Okay. That's perfect. Thanks for sharing that. Just last question. Do you have any advice for people that are either already in the field or trying to get in the field? Anything that you would recommend for people trying to, I don't want to say trying to make it, but just trying to innovate in the biosciences? Do you have any advice for them?
There's so much unexplored territory out there. I could show you, we have patents on the wall here that we haven't even started really using. we have so many ideas and technologies that we want to bring to reality, to the market, and it just takes such a long time to refine and improve and commercialize any one piece of technology that doing it over and over again, it's a lengthy task. And so there's no shortage of interesting ideas that need to be explored and need to be commercialized. So there's certainly lots of room. The question is, do you also have in place the things that are necessary to make your venture successful? And it's not just the technology idea, right? Mm-hmm. It's execution, making sure you have the finances, you have a business plan, so understanding how you're going to make money, even in the early days before the technology is fully commercialized. So all of those pieces need to be in place in order for a venture to be successful. So, there's also no shortage of advice on how to be successful as an entrepreneur. I don't claim to have a corner on that at all. But I would certainly say that getting as much exposure and understanding to the kind of pitfalls you might fall into, the kind of issues that you're gonna need to be dealing with, as much of that as possible is certainly would be good advice. I myself try to get a lot of experience under my belt before I jump ship, as you say, and getting experience both on the R&D side, on the marketing side, on the management side, on the IP side, and doing all of that has been just absolutely critical. Because if I didn't have those experiences, I would have been struggling even more than we did to get to where we are now.
Wow, no, that's great advice. I really appreciate it, and I think there's no better place to end it. Thank you, Giacomo, for your time. It's been great collaborating with you and your company. We look forward to continuing the collaboration this year and next year. And yeah, I appreciate your time and thank you so much.
It's my pleasure, Javier. I appreciate the opportunity, and it's been a pleasure for me and my team as well to work with alongside your engineers. Has been a very, very great learning experience, and we got a lot out of it.
Perfect. Thank you so much.
You're welcome. Thank you.
Thank you, Giacomo.